I'd like to thank the organisers for inviting me to speak at this meeting on the subject of long-range conductivity in non-redox active proteins. Almost all of the work in protein electronics to date has been done on proteins containing redox cofactors. We'll see in this talk that ordinary common or garden proteins lacking redox cofactors can make very valuable electronic components. My lab stumbled across the high conductance of non-redox active proteins about 8 years ago when we thought we would try an experiment to see if we could detect single proteins in a chip we had designed for nanopore DNA sequencing. That's illustrated in panel A here. To do this, we put a ligand that bound a protein integrin to the electrodes, and we thought perhaps we would see some subtle changes in capacitance when a protein bound. In fact, we saw enormous jumps in current on the order of a nanoamp, and these were shown by control experiments to be specific to binding events. I simply didn't believe these results at the time because everyone knows proteins are insulators, and in particular non-redox active proteins are surely insulators. So why must proteins be insulators? Here is an expression for the conductivity of a material in which you see that the two parameters that matter are n, the density of free carriers, and d, the carrier diffusion constant. Now, my knee-jerk reaction that proteins have to be insulators comes from the fact that n is 0 in a molecular solid where all the electrons are tied up in bonds. However, thinking about this a little more, of course one can make n non-zero by injecting carriers. So n is a non-intrinsic parameter that depends on the experimental method used to inject charge into the protein. What about d? Well, over long distances, transport is surely through a hopping mechanism, and that's described by the Marcus theory. So down here in the lower left of the slide, you see the picture of the free energy of a system with the charge on molecule 1 and molecule 2 uncharged, and then displaced by some amount 2 lambda in electrostatic energy, the charge on molecule 2 and molecule 1 uncharged. This is a plot of free energy because this transition is brought about by vibrations versus electrostatic energy. Now this displacement, the energy taken to charge the system, actually twice the energy taken to charge the system, is the so-called Stokes shift, a lambda. Now what Marcus showed was that given the fluctuation dissipation theorem, which says that the effective spring constant resisting the charging, the forced charging, is also the spring constant that is responsible for the thermal fluctuations, then the curvature of these parabolas is given also by this same parameter lambda, and then some very elementary algebra tells you that this barrier here, where the two states intersect in energy, is just lambda by 4. So what does this mean? Well, with proteins, the Stokes shift is typically around 0.8 volts. So lambda on 4 is 0.2 volts, or about 8 times kT. When you put this into the exponential expression for hopping rates and use the appropriate models for tunneling rates, the time for a single hop turns out to be a microsecond. Now, I just showed you data with nanoamp currents crossing a whole protein. Nanoamp currents means nanosecond timescales. So this process, this Marcus process, is 1,000 times too small. So what I mean by saying that the protein is an insulator is that even when you inject charges, the diffusion constant is too small. So now let's turn to experimental measurements. The key point to remember here is that no charge injection means no free carriers, means no conductivity. So we need to be able to make contacts between electrodes and proteins that allow for charge injection. Our tool for exploring this is the electrochemical scanning tunneling microscope, shown schematically here in panel A, where a conducting probe is held a few nanometers above a conducting substrate, and a protein bridges the two via ligands that are bound to the metal electrodes and then in turn capture the protein. The whole experiment is submerged in electrolyte and under electrochemical potential control. Now this makes physicists' hair set on fire because we are talking about electron transport and we're operating in a salt solution. What's going on? The whole point is that by controlling the electrochemical conditions, we can completely avoid Faradaic currents. So electrochemistry is actually the best defined way of controlling these surfaces and having intact proteins on them. As I said, we used ligands to capture the protein. So for example, epitopes to capture antibodies on both the substrate and the tip. These have the benefit that when both are appropriately coated, the protein, the target protein, will bind selectively to them and not denature as it would do typically on a metal substrate that was untreated. Using this device, we then take current voltage curves, and they look like the curves shown in panel C. And what you see there is no current when there is no specific contact to the protein. That's the curve labeled control. But once the target protein is bound, you see a beautiful linear current voltage curve, quite reproducible on sweep up and sweep down. I'll mention these spikes that I've labeled TN later on. Now we take many hundreds or thousands of such measurements across a substrate to obtain a distribution of conductances, and these are shown plotted on a log scale of conductance in the lower right of this panel. A key point here is that when we make one specific contact to an antibody, for example an FAB fragment which is bound by the epitope to either just the substrate or the tip, we see one peak in this distribution. However, when we use a full antibody fragment so that we can now bind at two sites, we see one peak at the lower conductance for a single contact and another peak at about 10 times the conductance for a double contact. And this is important because this means that the current must be going into one arm of the antibody and out of the other. And this distribution has been shown now for many proteins. And in summary, no specific contacts, no conductance. One specific contact, one conductance peak. Two specific contacts, two conductance peaks. And sometimes things like surface cysteines will give rise to another conductance peak. The implication is that if the large conductance arises from two specific contacts, the current goes in, for example, one head of the antibody and out of the other, which is a path length of 13 nanometers. Is this really what's happening? These plots here on the left of the slide show conductance distributions taken as the gap is systematically increased here by a total of 3 nanometers. And what you see is something absolutely remarkable. The number of counts one obtains in an experiment goes down as you make the gap bigger and disappears altogether when you make the gap much bigger than the original set point plus 4 nanometers. But the position of this conductance peak does not change. This is remarkable. It's saying that the conductance is independent of the gap size over a very large range of distances. This can only be the case if the path is through the protein, which is merely changing its angle of attachment as the tip and substrate are separated. I want to mention briefly this telegraph noise because it looks like Morse code when you sit at 1 voltage and look at the current as a function of time. I want to mention this telegraph noise again, because we noticed that in all the proteins we looked at, it onset at about 100 millivolts. So this plot on the right, lower right here, is with the DC part of the curve extracted and several hundred curves averaged, and you clearly see that the telegraph noise disappears below 100 millivolts. It was looking at this that made me realize that if one operated these proteins at a bias less than 100 millivolts, then any noise one saw must come not from this contact noise, but rather from conformational fluctuations of the protein alone. Here is an illustration of that phenomenon for an important enzyme, the DNA polymerase enzyme. This exists in two states, an open state where the DNA chain is ready to be extended, and a closed state where a new base is being incorporated. One can permanently close this complex by using so-called non-hydrolyzable nucleotide triphosphates. So therefore we can use molecules which are either permanently open or permanently closed and measure the distribution of conductances. In the upper panel, you see the conductances for a set of permanently open molecules. There are actually two specific binding peaks, the green and blue, corresponding to two types of contacts that can be made specifically. When we look at the closed form of the polymerase, we see that these peaks move all the way out to about 3 times the conductance in the open state. So there is a large effect of conformation on the conductance of an enzyme. Can one measure that dynamically? And the answer is yes, one can. These are polymerase molecules trapped in the STM junction and fed different DNA templates to copy. The template on the left is one that the polymerase doesn't much like and stops and starts and stutters and has trouble with. The template on the right is one that is processed in a very nice uniform way by the polymerase. And you can see indications of both types of behavior in the time resolved signal recorded from the enzyme. And it's pretty certain that each of these spikes represents a nucleotide incorporation into the polymer. And clearly we can distinguish different polymers through their characteristic signals. Next, I want to turn to a quantitative investigation of how current decays with the length of the protein. To make this investigation, we synthesized a series of linear proteins, here labeled CTPR, all the way from 4 nanometers out to 20 nanometers, and they had cysteines at each end of the protein, so we could attach them to a tip and a substrate. We made measurements of the distributions as I showed you before, and in the next slide we show how the distributions change with protein length. The red points on this plot show the positions of the peaks and the conductance distributions for each of these various proteins. Now, what do I mean by saying that a protein is an excellent conductor? Well, I mean relative to molecular wires synthesized to be excellent conductors. The black points on this plot are for oligophenylene imide, a famous molecular wire. And what you see is that while the conductance is higher at short distances, this molecule has a lower contact resistance than the protein. The fall off with distance is much steeper than for the protein, with the result that after about 5 nanometers, this common or garden non-redox active protein is a better molecular wire than a molecular wire designed to be a wire. And that's what I mean by good conductive properties. One last experimental aspect I want to address is the energy of the charge carrying states in these proteins. We were able to probe this by changing the energy of electron injection in several different ways. The data in this slide show experiments in which we used different metals for the tip and the substrate, so that the average Fermi energy was then moved around as a function of the metal combinations used. Now this plot here shows the conductance versus the average Fermi energy, but expressed here on the normal hydrogen electrode scale for electrochemistry instead of the usual vacuum scale. The reason for that is we repeated this experiment by using fixed types of metal and then changing the surface potential electrochemically, and in that case one reads the potential out directly on this normal hydrogen electrode scale. And a third way was putting charged entities at the interface with the electrodes. All three ways gave the same result, that there was a peak in the conductance at about 300 millivolts on the normal hydrogen electrode scale. Now this is surprising. Why? If you think the mechanism is hopping by the creation of a hole, i.e. the oxidation of an aromatic residue, the energy to do this for tyrosine and tryptophan is well known. It's about 1 volt on the normal hydrogen electrode scale. Our charge carriers have somehow managed to lose about 700 millivolts of creation energy. Where has this gone? What are these states? That's what we address next. Turn now to the recently published calculations carried out numerically by Alec Aksamentiev and Siddharth Krishnan at UIUC under the guidance of my colleague Dmitry Matyashov at ASU. These all-atom simulations had quite a striking result. They showed that charge transfer is very rapid between the aromatics, and that this means that the reorganization energy is reduced because the system is not fully relaxed. This explains both the long range of conductance and the reduced oxidation potential. Let's take a look at how these calculations are carried out. So let's take a look under the hood of these calculations by focusing on just one pair of aromatics in the protein. Of course, the calculation is carried out for all pairs between which charge transfer is possible, but this pair will illustrate the way in which the calculation goes. Panels A and B show the canonical Marcus pair, in this case of tyrosine 77 in panel A neutral and tyrosine 43 positive, and then in panel B tyrosine 77 positive and tyrosine 43 neutral. The electrostatic energies calculated in water with counterions present are shown in the plot in panel C. As you can see, the mean electrostatic energy is quite constant for the two states, and the separation of those two black lines is around a volt, meaning a Stokes shift of about half a volt, but the fluctuations are substantial. The lowest energy of the red points overlaps the highest energy of the blue points, and it's these overlap points that are the transition energies for tunneling. If we take this energy versus time and turn it into a probability distribution, we get the Gaussians shown in panel D. Gaussian because of the central limit theorem. These Gaussians in turn can be turned into the free energy parabolas to get a Marcus-like plot by using the Boltzmann equation. And when one takes the logarithm of the Gaussians, one gets a parabola shown in panel E, so there's the pair of Marcus parabolas. However, what is not Marcus-like are the numbers. The Stokes shift is half a volt, and for half a volt one would expect a barrier of a quarter of that, or 125 millivolts, about 5 kT, but you can see directly from the plot that the barrier is only 50 millivolts. The reason for this is that the actual curvature parameter, lambda, which dictates the width of the parabolas, is much larger than the width you would get from the Stokes shift, about a volt compared with half a volt from the Stokes shift. And so that means the parabolas are broader and intersect at a lower point. So here's a summary of results for all pairs of aromatics that lie within about 1.5 nanometers of one another for this particular protein. And the parameter lambda r here is the reaction reorganization energy. You saw the barrier was 50 millivolts. This is given by lambda Stokes squared on lambda. This follows from the geometry of the parabolas. And so with the 50 millivolts we had before, that corresponds to a reaction lambda of about 200 millivolts. And you can see that's actually pretty common for most of these transitions with a fairly broad distribution. When these energies are used to compute the barrier to hopping, one can then, from a fairly standard model, calculate the time scale of the hops, knowing the distance between the edges of the aromatic residues. And that's the distribution here in panel D. And you can see that most of the hops are sub-nanosecond, as opposed to the microsecond that we expected from Marcus theory. And sub-nanosecond is clearly what we need in order for electrons to traverse a protein at nanoamp currents. Turning now to the energy of the states involved in hopping, the injection energy at the terminals will represent the difference between the Fermi energy of the metals and the free energy of formation of an oxidized state, delta G0. Now, in the case where the movement of charge is so rapid that the state is not fully relaxed, Dmitry Matyashov has shown that the free energy effective free energy is reduced by an amount of lambda Stokes divided by lambda. And as you saw, that lambda Stokes was on the order of half a volt and lambda on the order of a volt. In fact, on average for all these transitions, the factor is about 0.4. And so for a 1 volt delta G0, the actual non-equilibrium oxidation potential will be around about 400 millivolts with delta G0 1 volt. This is somewhat close to the 300 millivolt peak that we observe in our experiments. And so you see the non-ergodic nature of the charge transfer events also explains the low energy of formation required for these states. To get at the distance dependence of the current, we're going to need a diffusion constant. However, we have the time for hopping transitions calculated as I just described, and of course we know the distance between the pair associated in a particular transition, and therefore we can heuristically define a diffusion constant for each particular hop and then average over all the hops over all possible pathways. This is a heuristic answer to the problem because it turns out that an exact analytical solution for a random distribution of spatial distances and a random distribution of times has not been solved. Nonetheless, with this heuristic in place, we can Monte Carlo average the diffusion constants over the pathways. And in the case of this particular protein, we had 2 aromatics near one of the contact cysteines. So we did it for both sets of pathways, and the result was about the same. That was a diffusion constant of about 20 nanometers squared per nanosecond. What's very interesting is this is the result that was found by an Israeli group, or the required value of diffusion constant found by an Israeli group to account for the remarkable conductance of the OMCS wires studied by Nikhil Malvankar. So this ordinary non-redox active protein has a diffusion constant that is very similar to that required to explain the incredible long-range conductance of OMCS. So the last missing piece of the puzzle is the density of free carriers in the protein, and to find this we need a model for charge injection. Dmitry modeled this as electrochemical injection from the metal electrode using a coupling parameter that has been determined from electrochemical data for a redox protein. adjusted a little bit to fit our data, but nonetheless close to a value that is accepted in the literature of a few microvolts. So with this one adjustable parameter then, which once again is contained within sensible limits from electrochemical literature, we can then model charge current density injected. We can solve for the carrier distribution using our calculated diffusion constant, and knowing the injection rate and the extraction rate, we can calculate a net current in the protein at a given bias. The red line in this plot is the theoretical calculation. It's nonlinear with length because of potential drops at the contacts, whereas the black squares here are the experimentally measured data. And with this one adjustment of the coupling constant delta here, the data are quite well fit by the theory. So I want to end by saying a little bit about what this might be good for. I've already alluded to DNA sequencing and detection, for example, of single antibodies. We have a small grant from the NIH to look at current fluctuations in a proteasome as individual proteins are digested, with perhaps the possibility of sequencing proteins at the single molecule level. We've done a little bit of work with CRISPR-Cas9 to detect transitions when it recognizes its substrate. And finally, we also have a small grant to study the digestion of glycans by heparinase with the goal of sequencing linear glycans. So I think it's a potential platform technology. So in summary, I've shown you that many proteins lacking any redox cofactors are excellent conductors, meaning nano-Siemens scale conductance, if they are chemically connected to electrodes and not denatured. Charge injection and transport are made possible in these systems because of the non-ergodic transport, which reduces barriers to hopping to the order of kT in energy, and this is because the hopping time is fast in these circumstances, a fraction of a nanosecond. At low bias, current is relatively free of contact noise, and this means that conformational fluctuations dominate current fluctuations, meaning that the dynamics of enzymes can be followed on a single molecule basis. There are many possibilities then for nanoscale electronic devices with the chemical versatility of enzymes, the self-assembly properties of proteins, and the tunability of properties enabled by recombinant engineering. And finally, none of this would be possible without the help of many folks in my lab and collaborators. Particularly, I want to acknowledge Bintian Zhang, who really did the bulk of work on this project, and the folks who funded us patiently for a number of years.